for us as people who are interested in kind of the social implications of genetics, partially because of this issue. It used to be that if you did a genetic test, it would tell you you have cystic fibrosis or you don't, you have muscular dystrophy or you don't. And those tests still exist, but more and more the kinds of genetic tests that we're doing have much higher levels of uncertainty, right? With information that tells you you may be more susceptible to particular cancers, you may be more susceptible to heart disease, right? And it raises all sorts of questions for individuals and families because, you know, we like certainty. We like to know when you go to the doctor that the doctor says you are going to have this or you're not going to have this. And that's just not the case with most of the genetic information that we're getting now. It raises your risk. But we're, I think, as humans, not so great at thinking about risk. Even doctors that we work with are not so great at thinking about risk. If someone tells me that I'm at a 5% increased risk for heart disease, I don't really know what that means, and it might make me a lot more worried than I need to be. If someone tells me that I'm at 80% more risk for heart disease, I still might be more worried about it, but I'm not exactly sure what that means. And so there's a much higher level of uncertainty with the kinds of genetic testing that many clinicians are starting to use in clinical settings. It also may depend on your background, right? So Ashkenazi Jews, African Americans may have different kinds of risk levels depending on their geographic ancestry. And this is also kind of raising all sorts of questions. As I'm sure many of you have started to see on TV, maybe some of you have done this, is that we're also starting to see genetics not just being handled in the clinical settings. We're starting to see genetics being out in the public, being able to be gifted for Christmas, for Hanukkah, for New Year's, for birthdays, right? How many of you have seen advertisements on TV for 23andMe or for Ancestry.com, right? It's all over the place. In fact, 23andMe last year was listed as the number 5 Christmas present in the country, right? It's becoming part of our everyday lives. And, you know, for some individuals, getting their ancestry testing or 23andMe results are fun. They're interesting. It's recreational. It's something that you might want to do because you want to learn more about your background, your history. And that's, I think, a good thing. We I think, should be encouraged to learn about ourselves. 23andMe is an interesting case because 23andMe gives you information about your ancestry. It also gives you information about what we might call fun genes or fun genetic information. So genetic traits that might make you taste cilantro differently than other people, right? Some people taste cilantro and it just tastes like cilantro. Some people taste cilantro and it tastes like soap, right? Some people hate cilantro, right? And there's a genetic change that indicates whether or not you can taste cilantro or not. If your face flushes after drinking alcohol, that is a genetic trait that can be seen in the 23andMe. A little gross for Sunday morning, but if your urine smells after you eat asparagus is a genetic trait that is listed on 23andMe that you can find, right? And those kind of things are kind of interesting, kind of fun to learn about. But there's another side of it, which 23andMe also gives you information about your health, about your risks for different diseases. And that may be really good for you. It may also be confusing. It may lead to you being worried about your health in ways that maybe you don't need to be. It may lead to you, not really knowing whether or not you should change your lifestyle. And so, doctors are increasingly having patients come in and saying, hey, I got this 23andMe result. And what should I do with the information? And I can tell you most doctors don't really know what to do with the information because they were not trained in genetics the way that most medical geneticists are. Luckily, we do have professions in the United States and really worldwide that can help you. We have medical geneticists. We have genetic counselors who are just unbelievably prepared to help families to go through and look at their results and understand the results. But not everyone has access to genetic counselors, right? If you have poor access to healthcare, if you're in an underserved community, a health disparities community, you may not be able to go and see a genetic counselor to hear about this. So there's some dark sides to having this kind of information. There's also the dark side to ancestry, right? We see commercials on TV of, oh, well, I thought I was Irish, but I'm actually German, and that just meant that I just changed my kilt for lederhosen, right? That's a fun story, right? But for some people, this can be devastating information, right? We're doing a study right now where we're looking at stories of families who said, I got 23andMe for Christmas, and it ruined our family's Christmas, right? Because people find out that who they thought they were not who they were. And that may mean your background may be different. That may also mean that the people who you're related to may not actually be related to you. People through this information can find what's called misidentified paternity, which means dad wasn't dad. And that happens not all the time, but frequently enough that it makes us worried, right? There's a story we were tracking about a boyfriend and a girlfriend in college in Tennessee who got each other for Christmas the 23andMe kits, and it turned out after they got their results that they were siblings. Because they were from the same fertility clinic in a small town and didn't know this until they did their 23andMe results, right? So we need to think deeply about how these kinds of genetic tests impact the way we are to each other, our identity. The other thing that Marsha was talking about right before we started is that 23andMe uses your data. Right? So it's so cheap because they want the data from you, and they sell it to pharmaceutical companies and other kinds of places. And so when you sign up for 23andMe, you sign a consent form that says any data can be used in commercial settings and in other settings. And that is also concerning for folks like us because we worry about the privacy of people's information. Right? And all of this kind of leads to where I'll end, which is kind of some big questions about ethics, right? So here's a couple different questions that we want to pose to you to get your brains kind of thinking, and then we'll have some time to discuss after, right? So, well, I just messed that up, sorry. Right, so do I have the right to know my own genetics? I think we would all say, yeah, it's my genetics, I should have a right to know it. But right now, the technology isn't really good enough to be able to just go to your doctor and say, I want a whole genome test, right? And that eventually will happen, but it's not quite here yet. Do I have the right to know my children's genetics? This is a real hot topic right now because people might say, oh yeah, they're my children, I should be able to know their genetics. But what about knowing about diseases that your child might get as an adult? Should you be able to know that information, or should it be up to your child to make that decision of whether or not to know when they get older and can make a decision to get genetic testing themselves? Who will have access to my information, right? The idea of genetic discrimination has been around really since the beginning of genetic testing. Will people be able to discriminate against me based on my genetic information, my employer or my insurance company? We do have laws in this country, including one federal law called GINA, the Genetic Information Non-Discrimination Act, that protects individuals' genetic information from being used against them in employer or insurance settings. But even that law is not perfect. If you work for a very small company that has under 50 individuals, you're not protected by GINA. If you are looking for long-term disability insurance or life insurance, not protected by that law, and so you can still be discriminated against. If you work for the armed forces, you're not protected. So there's a number of places where, yes, you're protected, but not always, right? You're not always protected in every situation. So we need to think broadly about how and who has access to our information. So this is a different question, which is who has access to genetic testing. Genetic testing is still very expensive. If I want a whole genome sequence, it's still somewhere between $2,000 and $3,000, right? There are lots of people in this city and in this country who can't afford $3,000 for a test, let alone $1,000 or even sometimes $100 for a test. And so we also have to think about equity and justice questions when we start thinking about genetics, and how do we make sure that all individuals can get access to genetic testing and genetic services. And then where I'll leave you and where Marsha will pick up, which is how should genetics be used to prevent diseases in future generations? What characteristics were coming from each plant, and if anyone remembers from their high school genetics classes, those Punnett squares where you had to fill out the X's and Y's, really all come from Gregor Mendel. It really was the start of modern genetics. It wasn't till about 100 years later that we truly understood the structure of DNA, that DNA was the mechanism by which heredity was passed on from parents to their children, from children to grandchildren and future generations. In the 1950s, 1953 to be exact, the structure of DNA was discovered by Watson and Crick at Oxford. And I also have, so this is a picture of Watson and Crick with their first model of the double helix of DNA. I also, very importantly, have a picture up here of Rosalind Franklin. She was the X-ray crystallographer who took the actual picture of DNA, the first actual pictures of DNA, and was largely, almost exclusively left out of any announcements, and was not granted the Nobel Prize. When Watson and Crick granted the Nobel Prize, was notoriously discriminated against in their lab. When asked how she could improve her work, was told by many people in the lab, including Watson and Crick, that she should hike up her skirt, and that would be the way. And she is kind of one of our heroes in genetics for the work that she did, and that she really persevered through her work and is now being recognized. Unfortunately, she passed away before the Nobel Prize could have been given to her, and the Nobel Prize is not given to people who have passed away. But more and more, when you hear about the kind of history of genetics, Rosalind Franklin's name is inherent to that story, and I just wanted to make sure I brought her into the story. In the 1970s, we started to see the first ability to put pieces of DNA into other pieces of DNA, what we call recombinant DNA technologies. Much of what Marsha is going to be talking about in a little bit stems from the work in the 1970s, where we were able to kind of do recombinant DNA work for the first time. It's also the first time that ethicists like ourselves got very worried about genetics. Right, we started to worry about maybe we're starting to manipulate DNA. Maybe that raises questions about ethics and about humanity and about who we are as people. That led into the 1980s, where we had human gene identification and mutation analysis. This was really the first time that we were able to, using samples from humans, look at gene changes, small gene changes that might lead to diseases. And you can see here, there's a picture of a southern blot. This is an ability to look at different base pair changes. You know, our DNA is made up of A's and T's and G's and C's, little chemical base pairs. This was the first time in the 1980s that we were able to kind of go in and look at those genetic changes and see what kinds of genetic changes might lead to diseases. In the early 1990s, it was announced that the US government was going to try to sequence the entire human genome, read all of the A's, T's, G's, and C's, 3 billion base pairs of the human genome. People thought it was going to take somewhere between 20 and 30 years to do that, but changing technologies got it done in about 13 years. So in the 1990s, we were able to map all the chromosomes, the human chromosomes, and look in and read, just like a book or a phone book, we were able to read all those A's, T's, and G's, leading to 2003, where the first kind of full human genome was published. And now, ever since 2003, we're in what many people call the post-genomic era. We now have the human genome. We can now map the human genome, and therefore we can create genetic tests that go into your DNA and look for genetic changes that may do things like lead to why I'm only 5'4", and also why I might be more susceptible to heart disease or more susceptible to cancers. So, you know, it's important to remember, and this may be not new to all of you, that we're all 99.9% the same. Our DNA is incredibly similar to each other. We're actually over 99% similar to a chimpanzee. This is, our DNA is so similar, but there are about 1.1% differences between all of us, and it's in those 0.1% differences that we see all the things that make us look different, give us different diseases, different resistance to disease. And so genetic testing is really interested in that 0.1%, right? Genetic testing wants to go in and see, okay, this person individually, how does their DNA differ than others that might lead to disease susceptibility or blue eyes or brown eyes, those kinds of things. So genetic testing is really about understanding the differences that we all share in our DNA. We all have differences in our DNA. We like to say there's no such thing as a boring genome, right? None of us have the exact same genomes. We all have different traits. We all have different things that will impact who we are. And, it's important to also understand that we have genetic testing that spans the whole life course. Right, so we have genetic testing that can be done prenatally on fetuses to look for potential genetic abnormalities. We have genetic testing that's done on newborns. So I do a lot of work in newborn screening. So these are tests, and if you have kids, you probably remember that when your children were born, they got a little heel prick, on the bottom of their heel. That was a genetic test to look for genetic diseases that might manifest in early childhood. We have genetic testing that impacts pediatrics, that kids can get, all the way through older Americans, right? So really, genetic testing spans the life course. There isn't one age that there are more genetic tests than others. Really, we use genetic testing at all phases. And increasingly, we're using genetic testing in clinical settings for normal, everyday, average care. Just before this talk, we were talking about something called pharmacogenetics. This is genetic testing to look at how you metabolize drugs. So you may go in and need a blood thinner. Your doctor might order a genetic test to find out which blood thinner might work better for you, or how much to give you to give you a better dose. We're starting to use genetics to do testing on treatment plans for things like hypertension, right? Hypertension is actually a very hard disease to treat. It can be treated using diet, it can be treated using beta blockers, ACE inhibitors, diuretics, all sorts of different drugs can be used. And it's a little bit of a guessing game. You might go into your doctor and say, I have high blood pressure. They might say, let's try a beta blocker. That might work for a little bit. You might come back, didn't really work, let's try something else. Genetic testing is now leading to an ability to go into your doctor and say, okay, we're gonna do a genetic test, and we know that for you an ACE inhibitor is gonna work best. And this dose of an ACE inhibitor will work best, right? It's what we call personalized medicine. And that is very quickly kind of coming down the pike, right? It's really something that we're gonna start seeing more and more in our regular clinical encounters. And that's great, right? We wanna see that. We wanna be able to get, use genetics to give us better medications or to give us better information. It also leads to all sorts of ethics questions that we're gonna talk about today. I also want to say that, the kinds of tests that we're doing are changing rapidly. It used to be that genetic tests were almost exclusively like that first picture, where you're looking for one small change in your DNA, right? One letter difference or a few letters that are different that can lead to your inability to metabolize sugars, or that can lead to a breakdown of muscle tissue, right? And we still have tests that look at those things today. Tests for things like cystic fibrosis still are what we might consider single or small numbers of gene testing. But now what we're doing is what's called whole genome sequencing or whole exome sequencing, right? This is the ability to look across your entire genome to look for lots of different changes, lots of different things that might lead to disease or lead to susceptibility for disease. And that's really changing the game for us as ethicists and for you all as patients and interested community members, because it's not really just one test anymore. It's a test that can test thousands, if not millions, of genetic changes at the same time. And that, again, raises all sorts of ethical questions. Right? We talked a little bit earlier about the idea that we can do prenatal genetic testing, so looking at diseases that our future children may have. But what do we do with that information? How do we use that information? Do we use that information to prepare to have a child with a disability? Is that information used to make decisions about pregnancy termination? Or is that information used to make determinations about genetic engineering that might be able to prevent future diseases? So I'm going to stop and I'm going to hand it over to my colleague, and we're going to talk about some of the really, really hot topics that are going on right now.
And keep all these questions in mind, because even for these future technologies that I'm going to be talking about, these questions still come into play. They've been here pretty much since the beginning and will continue to be here. So I'm going to move us sort of into the future that is in some ways today. Oops, that's not the right one. That wasn't just me. Okay, so has anybody heard of gene editing? All right, what is it? Is it as easy as Microsoft Word cutting and pasting? So, I should use this thing since I can't seem to use the keyboard. So, here's how gene editing works. You can take molecular or bacterial tools that can cut DNA in specific places, and you have techniques for sort of programming them ahead of time for where they're going to cut the DNA. And then the cell's own DNA repair mechanism, which is always in operation, will step in and fill in those spots. And that will cause a deletion or an insertion or a change in that spot that could fix something that you're trying to fix or change something that you're trying to change. I like that picture, it's really cool. Is gene editing brand new? Nope. It's been around for a while. So we've had technologies in the already in existence, mainly called zinc fingers or talons, that were available to do this kind of work. But it seems like we've really only started hearing about gene modification or gene editing just in the past few years. So what's different now? What's different now is that we have a new technology called Crispr. Crispr stands for, you'll never need to know this in your entire life, clustered randomly interspaced palindromic repeats. And what the discovery is that there are some bacteria out there. The first one that was found was called Cas9, and that bacteria has its own innate ability to remember particular little sequences of DNA or RNA and then go in and cut at that spot. And it's a protective mechanism for the bacteria. And what we've been able to do, I say we, I don't do this. What scientists have been able to do is go in and sort of program the Cas9 or other protein or other bacteria to go in and look for specific sequences and cut at that spot. And then again, the cell's own DNA repair mechanism steps in and replaces that. Here's what's really important. CRISPR is cheap and it's easy. You can literally buy a kit online, have it mailed to your house, and do this in your garage. That was not the case before. It just took a lot of laboratory equipment, a lot of training, a lot of money to do these things before. CRISPR is very, very different. And I'll recommend, I've only seen the first episode, so I can't say about the whole thing, but there is a series on Netflix now called Unnatural Selection that's all about CRISPR, and it actually shows a guy in his garage trying to do CRISPR modifications on his dogs. And he ordered the kit online. So this question was in the brochure, and I wanted to make sure I addressed it. Can modified genes be passed on to future generations? What do you guys think? Yeah. Well, the answer is maybe, so you're partially right. So in humans, maybe, or maybe not. There's two different kinds of genes that we have. You can classify genes in a lot of different ways, but one way that you can classify them is somatic genes versus germline genes. Germline genes are the ones that get passed on to the next generation. Somatic genes do not. Somatic gene editing would be in a child, even a fetus, or a child or an adult, and those changes could not be passed on to future generations, as opposed to germline gene editing, which would be passed on via your eggs or sperm to future generations. And those changes can be inherited. So let's talk a little bit about somatic gene editing first, because this one doesn't get nearly as much press. I got a lot of love for somatic gene editing because nobody ever hears about it. But in fact, you probably may have heard about it just recently because there's been some really interesting stories. We've seen just in the past year to 18 months, some clinical trials go in that are actually using CRISPR in somatic modifications. A couple are for cancer. One is for an inherited form of blindness. And one is for sickle cell. And these are really interesting trials. They're doing super interesting work. And to be perfectly honest, very few ethicists are saying that there's really any big problems with this. Now, we do have some concerns about broad applications of somatic gene editing. These particular trials have been very carefully done, and I think most people would say it's really great if people who have an inherited form of blindness would like to get this treatment and it works for them. Now, germline gene editing, this is the one that everybody hears about. That really sounds really scary, right? And one of the reasons you're hearing about it a lot right now is because of this guy, He Jiankui in China. He is not even a geneticist. He's a biophysicist. And he decided, hey, wouldn't it be great to edit some genes and have some babies get born? And that's what he did. And reportedly there have been 3 babies now born from this experiment that he conducted. And he was just, last week it was just announced that he has been sentenced to 3 years in prison for deceiving the regulatory agencies and the universities that he was working with, and not telling them exactly what he was doing. So we'll see what kind of chilling effect this has on future sorts of rogue scientists. But germline gene editing on the whole, we have, of course, lots of questions about it, lots of concerns, but it hasn't been completely ruled out. There is certainly research that's being done on germline gene editing, and you can imagine that there might be some things that you would want to eliminate from your bloodline altogether, especially if you have in your family certain kinds of really, really serious heritable diseases. Now, here's the third category. Can modified genes be passed on to future generations? In non-humans, that's actually the reason why they do it. So in agricultural crops, domestic animals, wild plants and animals, gene drives, these are all situations in which gene modification is going on right now, and the whole goal is to modify genes that will be passed on to future generations. In corn, for example, in domestic animals like cows, in wild plants and animals like mosquitoes, right? Would you like to live in a world without mosquitoes? Well, maybe you can. Would you like to live in a world where rats don't pass on the kinds of diseases that they do now? Maybe you can. It would mean changing the genome, and in some cases doing what's called a gene drive, which is a specified kind of gene change that sort of pushes those changes to be spread faster than they normally otherwise would. And there's certainly research going on in all these areas, and sometimes it's already in the food supply, certainly in some agricultural crops. It's already in the food that we're eating. So, are we worried? Well, yeah, we're worried. So what are we worried about? We're worried about some of the same things that Erin was just talking about, for sure. But let's just talk about it in terms of these 3 different categories. So somatic gene editing, yeah, there are some concerns. We have safety concerns, for example. CRISPR's not perfect. CRISPR the bacteria can make SNPs in the wrong place. And when that happens, those insertions could cause more problems than they solve. It could cause cancer, it could cause all kinds of things. And the other thing is listening to patients and families about what they in fact want. So we have a colleague, Vince Bonham at the NIH, who is doing research with families that are affected by sickle cell anemia. And he's getting a lot of great feedback from them about how they feel about CRISPR and gene editing for sickle cell, mostly positive. I've done some research with my colleagues about families of people with Down syndrome and how they feel about potential for gene editing to eliminate that condition. And there's a lot more ambivalence, let me tell you. Some people think it would be the best thing ever. Some people think, absolutely not. I love my child exactly the way they are, and I don't want you to eliminate them. Some people are very ambivalent. They say, you know what, I love my kid, but if I could make life easier for them, I would. And I worry about what's going to happen after I'm gone. So there's a lot of ambivalence. And when you think about conditions that maybe are even more sort of perceived in very different ways by different people than Down syndrome, something like autism, for example. You imagine if you asked a community of people with autism and their families how they would feel about gene editing to rid them of this condition forever. You might get very, very different responses. So we definitely want to listen to patients and families about what their experience is and what they actually want. Germline gene editing obviously creates a lot of concerns, and you can imagine what some of these are. One is that it's irreversible. It's really hard to put the genie back in the bottle. Once you've got this trait and it's heritable, then there it is. How do you get rid of it? We worry about the safety not just of the future offspring, but also of the mother. This is something that we're still discovering and finding out about. But when you're pregnant, some of the cells from the fetus embed themselves in your system and can stay there forever. So if you have a gene-modified fetus, does that change your genome forever? What does that mean for your future reproductive capacity? If you're carrying a pregnancy and the gene editing somehow goes horribly wrong, what then happens? Are you able to have more children and not worry about that gene editing change affecting your system? Hard to know. I'm embarking on a research project on just this topic to find out what the risks are to especially women participants in these trials, because you don't get gene-edited babies without moms. Somebody has to carry those babies, and they're not just carriers, they're people and research participants and patients. And then, of course, who decides? Who decides what conditions are bad enough or important enough to edit? A lot of people think, when they think about gene editing, the first thing they think of is designer babies. Are we going to be able to design our babies? And who decides what's the best design? What's the primo kind of design for a baby? Is it blue eyes and blonde hair? Is it slender? Is it athletic? Is it smart? Is it different for men than for women? Do we want our women to be beautiful and our men to be smart? Vice versa? We have to decide these things if we're going to start designing babies. It's a little early yet. We can't actually do any of those things yet. We can figure out what color eyes babies are going to have. But beyond that, some of these traits are quite complicated. Erin was talking about height. There's a lot of genes implicated in height. There's a lot of gene-environment interactions. So certainly, we're not quite there yet, but we need to be thinking about it. And then finally, non-human gene editing should concern us all. Is it irreversible? Where does it proliferate? Does it get beyond my field of crops to your field of crops? Does it get out of Ohio to Kansas and Illinois and Tennessee, right? Where is it going to go? Are we going to lose biodiversity if we edit the genes of a species in one area to eliminate a trait? Are we then going to lose the biodiversity that is somehow important for other things? And is that going to have effects on our whole biome and effects on us, frankly? We're going to be consuming these crops, we're going to be interacting with the biome. And then finally, what's on the horizon? So we're going to have continuing debates about germline editing and designer babies. And as I hinted before, we can't really design most of the things we might want to, not yet, but maybe in the future. We're going to see increasing use of somatic gene editing for serious disease. We're going to see massive use of gene editing in agriculture. It's already really, really increased a huge amount in the last 3 or 4 years, and it's going to keep increasing. We're going to see more citizen science and biohacking, like in that show I was talking about. And there's going to be lots of new ethical reports coming out. There's going to be reports from the WHO coming out this year, from our National Academies of Sciences. There's going to be policy statements being issued very soon by lots of other organizations. But we know that conflicts and gaps in international regulations are going to leave a lot of loopholes that we're going to have to address. So with that, thank you very much from both of us, and we're happy to answer lots of questions.
Updates of the next forum that's coming along. So, and we also have the forum brochures. If you did not get one last week, please take one, and we'll have more later on. And in terms of questions, remember that it's the person with the microphone that will choose the question. So we go back and forth, and that's about it. I'm going to start these now.
And so if you have a question, raise your hand and someone will bring you the microphone when it's your turn. And please hold the microphone up to your mouth and speak directly into it so we can all hear you. And make your questions brief, no speeches, please.
I have 2 very basic questions. One of them is, how does the CRISPR get into the gene? I mean, do you swallow it? Do you inject it? In your garage? What are you doing in your garage? And the other is, when the part is snipped out, what does the body replace it with? A modified gene or a...
Yeah, so you
Or
would
more of the
you would
same.
inject, and often inject a viral vector or a stem cell. So you might take out some of that person's own stem cells from their bone marrow or from their fatty tissue. You would modify it in vitro in the laboratory and then inject it back into them. And when you do that, you also inject some of the stuff that you want it to be replaced with. So the bacteria will snip out the part, and then it's looking for something to put in its place, and if you've injected some of the right stuff, you hope that that's what gets replaced. But if it sounds a little haphazard, yes, sometimes it is, and sometimes there's mistakes.
I was talking to a friend who, and I was asking, why does there seem to be so much of an upsurge in gluten intolerance these days that we never heard about? And she said that years ago, people just ate natural wheat, but there's been so much genetic modification that it's been changed, and it could cause problems in some people. Is that a fact?
We don't know, but there is genetically modified wheat on the market. One of the things that's happened is because wheat is so heavily, there's such a heavy use of pesticide with wheat, that there's modifications to wheat to make it what they call Roundup resistant, so that it's resistant to the right kinds of pesticides. And I don't know the answer. I don't think anybody, maybe outside of Monsanto, knows the full answer to that question. But it's possible that that's a reason. It's also possible that people are just paying more attention to those things like gluten intolerance these days than they once did. So it could be a combination.
Thank you for a very interesting presentation. One question. What are the different types of genetic testing? The only one I know about is diagnostic. And two, has genetic testing led ever to mandatory sterilization of women?
I get the tough questions. So I'll start with the types of genetic testing. Most genetic testing kind of comes in one of 2 or 3 forms. So there are tests that are more what we call the genetic screening, right? And those typically will look for genetic changes that might make you more susceptible to something else or not. They're not definitive kinds of genetic testing. There's predispositional genetic testing, which is diagnostic, but still in the realm of risk. So BRCA1 and BRCA2 testing, for example, these are genes that lead to higher risks for breast cancer. They're diagnostic in the sense that they will tell you whether or not you actually have the gene change, the gene variant. It can't tell you whether or not you absolutely have breast cancer. Right, so even families who have BRCA1 and 2 in their families still does not mean you will absolutely get breast cancer. The risks are much higher. There are some genetic tests that are more certain, right? So if you're doing genetic testing for something like Huntington's disease, if you're doing genetic testing for cystic fibrosis, Duchenne's muscular dystrophy, those are diagnostic tests where they really are more disease-oriented. Are you going to have a disease or not? Most of the genetic tests that are done for newborns are for metabolic disorders where the genetic change says you will get a particular disease or not. But those are, we hear about them a lot because that's kind of the work that we work in, but it's not very common. And then the fourth category, so you have kind of screening, you have genetic diagnostic testing, you have predispositional testing, which is risk testing, and then pharmacogenetic testing. And those are the ones that are specifically not focused on disease states, but really about your ability to metabolize drugs. In the clinical setting, that's actually probably the one that's most used, the pharmacogenetic. It's been used for probably the last 10 to 15 years. It's becoming used more and more. Lots of clinicians like to do a genetic test before giving you a particular drug because they want to make sure that you're going to have a good reaction to that drug. The other place that it's used is quite frequently are in kids with kids or adults with syndromes where you really don't know what's going on. And so a lot of times there are genetic tests that may say, okay, we have a child who seems to have a lot of things going on. This seems to be a genetic condition. We're going to do an exome or a genome. We're going to look across their entire genome to see if we can find changes that might explain their condition. Anyone want to take a guess at how frequently, if I have a child, so say if I'm a pediatrician and I have a child with an unknown syndrome that I can't figure out what it is, and I do a genetic test, how frequently do you think, what percentage of time do you think that when I do a genome scan, do I find something that explains away what's going on? Anyone want to take a guess? 2%? 10? It's gotten better. It's actually about 25%, about a quarter of the time do we find some kind of gene change. But here's the kicker for us, and this is the kicker for geneticists, is that very, very few diseases are just caused by genetic changes, right? So many of the diseases we experience are caused by an environment, stressors, smoking, eating habits. What we don't have a very good handle on yet are those gene-environment interactions. We don't have a very good way of knowing that this person with this gene change in this particular environment will manifest disease while this person won't. We're starting to see that, right? So for example, people who have gene changes that lead them to upper respiratory infections or higher risk for upper respiratory infections. If you live in areas with higher levels of pollution, then you are more likely to have that gene change get triggered and actually cause upper respiratory infections. So there's a lot of effort right now to try to do more studies that do a better job of understanding kind of those gene-environment interactions and what that means for individuals. Your second question about sterilization, we could have done a whole talk, and maybe if you're interested, we could come back at a later time and do a whole talk about kind of the history of the Eugenics movement in the United States, and not just the United States, but worldwide, right? So in the somewhere between the late 1800s and the 1920s, 1930s, there was a movement in the United States called the Eugenics Movement, which was the idea of trying to improve genetic traits within populations through what they call positive moves, so things like promoting good traits, better baby pairs, fitter family contests, promoting marriages between people who had particular, what were considered good traits. There were also a series of negative eugenic movements where people who were deemed to have negative traits were either voluntarily or forcefully sterilized. Right? And this took place during the 1910s to 1930s in the United States. 31 states had sterilization laws on the books where individuals were deemed to have lesser traits, people who were seen to be less intelligent, people who were had other types of traits that were seen that were deemed to be not wanted. Even people who were promiscuous, people who were, if you look at some of the criminals, people who were in, at that time, what were considered asylums, but basically mental institutions, were forcefully sterilized. Because the idea was that if we could sterilize these individuals, they wouldn't pass on their negative traits. Now, we can have a whole conversation on what that means and who gets to decide, just like Marsha was talking about, who gets to decide what traits are good and bad. And you can imagine that people in power were the ones who were deciding who was going to be sterilized, and those were mostly white men who were making those decisions. And sterilization rates in underserved communities, poorer communities, racial and ethnic communities was far higher. The numbers are somewhere between 65 and 75,000 officially were sterilized during that time, although the numbers are probably a lot higher than that because institutions did not keep great records on who they were actually forcefully sterilizing. The Nazis came over and learned about eugenic techniques from the American eugenicists in the 1920s and 1930s, and that ultimately led to some of the policies of the Third Reich. After that happened, the eugenics movements quickly came out of style here, thankfully. But some of those sterilization laws remained on the books until the 1970s, 80s, and actually some states still have those laws on the books. They're not used today, but it's something that we as ethicists think about a lot, right? We don't want modern genetics to lead to another eugenics movement, and we worry daily about this potential. And so we have kind of a dark history with genetics. It's easy for me to show a picture of Mendel and talk about how wonderful his peas were, right? But we also have kind of a dark history with the ways we think about heredity in this country. And we need to be very careful about how we do that to make sure that doesn't happen again.
It's important to note, vis-a-vis your question, that genetic testing as we know it today is much later than the actual eugenics movement and those sterilization laws. And the sterilization laws that are on the books today are not based on the genetic tests that we have today. They're not linked one-to-one in that particular way. Those sterilization laws were for traits that were perceived to be heritable, which included things like promiscuity and criminality and feeble-mindedness, whatever the heck that means, right? So.
Thank you.
We worry about that, sure.
Very important question. Thank you.
Thank you.
Yes, Dr. Michie, in follow-up to your question about, or your answer about injections, where are these viral vectors and stem cells injected in the body? In muscle tissue, blood,
Depends
or something else?
Depends on where. So for blindness, it would be injected in your eye,
Okay.
right?
And then how did the gene therapies navigate to the point that they need to be to manifest the therapy?
Possible that might be above my pay grade to answer that, but generally what they have is what they call lentiviral vectors, and they inject them close to the site where they want them to be, and frequently what they'll do is take out the part of the virus that would cause a disease and put in the part that they want to transmit. There's a question back here, and she seems like she might know something about this. Is it, no? Okay, all right, so I'm just making sure. If there are any scientists in the room that would like to give a better answer about that, you're welcome to. Do you have a better answer than that?
No, but I know one of the concerns is the inability to find the right place, right? So one of the concerns is that you'll have cuts that happen in the wrong place because we're not as precise as we need to be. Back in the late 90s and early 2000s, when gene therapy was really kind of being first used, there were some problems where you were injecting gene therapy and it wasn't getting to the right place, and it was causing a number of issues for people, including some fatal.
I also thought of one more thing. If you're using stem cells rather than viral vectors, stem cells can proliferate and cause cancer. So if you get them in the wrong place, stem cells, that's what they do. They proliferate and they could cause tumors that would be problematic if they end up in the wrong place. So there's all kinds of concerns for safety, and we just put those all in one box as opposed to the other kinds of societal concerns that we have. But certainly we want to make sure that these procedures are safe before we're imposing them on people who may already be sick from a particular disease.
I had a question. Is the somatic gene modification research going on for diseases like Parkinson's and MS? And if so, how close are they to finding anything?
That is a great question. I cannot imagine that they're not. Those are diseases that have caused such devastation for a lot of people. So, and Parkinson's, we don't really have a good sense of the genetic basis of Parkinson's as far as I know. MS, much more so. So MS, I would guess, is much closer than Parkinson's. But do you know anything about that, Aaron?
Yeah, I do know that there are a number of studies going on for a variety of muscular diseases, and I think that some of them are looking at muscular dystrophies across the life course. I believe there's one on Parkinson's. I'm not 100% sure. But some of the same mechanisms they're looking at childhood diseases are also impacting individuals with MS. And so it wouldn't surprise me. There's one that's going on right now that looks very successful for a disease called spinal muscular atrophy, which is one of the most fatal, one of the most biggest killers of infants who have this disorder. It's a really devastating disorder where children lose muscular ability very quickly. Their brains are fine, their minds are fine, but they lose muscular ability very quickly and have to go on a ventilator very, very quickly. It's a really devastating disorder. There's been a few drugs that have come out over the last few years that treat spinal muscular atrophy that look really, really good. They're incredibly expensive drugs. I mean, drugs that are within the millions of dollars a year. And so there's a trial going on right now to look at gene therapy for spinal muscular atrophy. Those gene trials I've heard are also being similarly used in adults for different kind of muscular disorders. So I think there are things going on. There's a lot of hope for families that many of these gene changes, gene trials will actually lead to treatments for diseases that have largely gone untreated for many, many years. So this is the light side of some of this, is that I think we are moving towards really, really some pretty amazing drugs and some amazing technologies that are going to hopefully cure kids with these really just devastating disorders.
Thank you for a really fascinating presentation. I wanted to know if you have any data on how widespread the use of pharmacogenetic testing is among doctors for tailoring the treatments that they are prescribing for various conditions, and whether insurance generally covers these tests.
It's very widespread for some drugs, and for some drugs the FDA actually has recommended. For warfarin, for example, there's a FDA says you should have this genetic test, and then it is covered by insurance. There are newer pharmacogenomic tests that may not be covered by insurance yet, because of course they depend on proof of concept studies, clinical trials, all kinds of assessments like that. But for the ones that the FDA has said, yes, this absolutely is an important test to do, then it becomes pretty widely available and pretty widely covered. And I don't know how many drugs there's pharmacogenetic testing for, but there's a fair number now. It tends to be, as you said, blood thinners, cancer treatments, yeah. Cancer treatments is a big one. If you've unfortunately had a cancer diagnosis, especially like something like breast cancer, they will find out if you're HER2 negative. Sometimes you'll hear that, if you have particular genes, and they'll tailor your treatment for that.
I'm just wondering, in terms of all the genetic testing, where Alzheimer's predictability lies with either the testing or the ability to predict that might be a factor.
That's not an area that I've dealt a lot with, but I will tell you that a lot of the research that's going on about Down syndrome, people with Down syndrome have a heightened susceptibility to Alzheimer's, and so what's happened is that some of the research that's going on about Down syndrome is shedding light on Alzheimer's more generally, and has given some hope to people who don't have Down syndrome but have found out that they have a heightened susceptibility for Alzheimer's. We do know that there is one gene that causes early onset Alzheimer's, but that's a minority. For most of us, the risk is not that we're going to develop Alzheimer's in our 40s. It's that all of us have some risk of developing it sometime in our lives, and we don't really know all the genetic basis for that.
And I'll just add a note, which is kind of a caution for us when we think about the idea of using genetics to predict disease, is that in many of these cases, like breast cancer, like Alzheimer's, genetically caused disease tends to make up a very small percentage of those with that disease, right? So if you have BRCA1 or 2, you do have an 80% increased or more increased risk for breast cancer, but BRCA1 and 2 breast cancers tend to make up what is less than, I shouldn't say a number, but it's a very small percentage of all breast cancers in the United States. And so we tend to think of these genetic tests as really, really important in showing who's going to get a disease and who isn't. But we tend to sometimes over-exaggerate how much this genetic information can tell us about overall rates of disease in a community. Partially because so many of these diseases are caused by not just genetic changes, but environmental changes and pollution exposures and other kinds of exposures. We're starting to get better at a whole other field called epigenetics, which is the ability to better understand interactions not only between ourselves and our environment, but our ancestors in our environment, our family members in our environment. We're looking at, for example, gene changes between generations. So I know that the exposures that my grandparents experienced affected my parents and eventually me. And so there's a number of studies going on worldwide looking at gene-environment interactions in previous generations and its potential impact. There's a famous study that was done in the Netherlands looking at parents of kids who were born during the time of famine after World War. There's a time in Amsterdam and in the Netherlands, very, very significant famine. And what they found are that children born during that famine then had children later who were more susceptible to obesity, because they had gene changes that were triggered by their grandparents that made them conditioned to think they were in a time of famine. And so their gene changes combined with environment are leading to higher rates of obesity in the Netherlands now because of grandparents who experienced that famine post-World War II. Fascinating, really important information that we are just now scratching the surface in understanding how even exposures that our grandparents or great-grandparents experiences can affect us as individuals. Really, really amazing and hard to kind of fathom kinds of studies that are being done.
I have a son who's 32 years old, and he has a rare genetic disorder called Prader-Willi Syndrome. And he was diagnosed at around age 1.5 with genetic testing. At this point, I mean, gene therapy is not something we're going to pursue because his brain is already affected and it's not likely to be beneficial. But I'm going to be at a meeting next week of families in Ohio with Prader-Willi Syndrome, and there will be a lot of young families with new kids there. And so what is the process? Let's say you have a 1-year-old and you've been diagnosed with this or any other genetic disorder. How do you go about exploring gene therapy and what's the process?
Well, it's really, I was going to say that it's not really initiated by the family, but I'm going to put an asterisk by that. I mean, you know, if there's no gene therapy, there's no gene therapy, and it takes years and years to develop these things and to make sure they're safe enough to put into our children and our sisters and brothers and mothers and fathers. However, I will say that for certain kinds of rare conditions, and Prader-Willi certainly counts as one of those, there are family-led organizations that have really pushed research forward in significant ways, and SMA is one of those conditions for which that's been the case. They form biobanks of tissues that help researchers. They fund research from organizations, and then that actually pushes research forward in a way that it wouldn't have been pushed forward because if a disease is quite rare, maybe it's hard to find funding for it in other ways. Do you want to talk a little bit about that?
So I'll just say one thing, because I know we're running short. I have a couple other questions, and I'm happy to chat after. There's also an organization called NORD, which is the National Organization of Rare Diseases. They are very good at connecting families and groups like the one you're speaking of with trials. One of the things that's hard with rare diseases like Prader-Willi is that because it's a rare disease, not every academic center is going to have researchers that work on that, right? So you have to find centers that do this kind of work. NORD is an organization that helps to connect families and groups of families with, so maybe Case Western's not doing any work on this, which I think they actually are, but let's say they aren't. Maybe they say, well, Ohio State is, or Michigan is, and they do a good job in trying to connect families and groups of families with researchers who sometimes researchers don't know where to go to find families, especially for rare diseases. It's very hard. You need those grassroots advocacy organizations to help make those connections. So happy to chat after, but as well.
My father died of Lou Gehrig's disease, ALS, 60 years ago. They knew virtually nothing about it then, and they don't know much more about it now. How much research is there on ALS, or this following up on Susan's question about Alzheimer's disease, or other neurological diseases?
I don't know anything about research on ALS specifically. I will say that what they're finding is that neurological diseases like ALS, Parkinson's, and other kinds of things like that, degenerative neurological diseases, even if they at their base might be caused by some different gene changes, they have a lot in common in the ways that those gene changes then act on the body. And so sometimes treatments that are discovered for one neurological condition can be transferred to other ones, even if the gene change itself is not the same. And so that's one of the reasons why we say, don't just think that it's all about the genes. It's important. Genes are really important. But what genes do is they produce proteins, and those proteins then act on your system. And sometimes 2 different gene changes can produce very similar changes. That's the case in cystic fibrosis. Cystic fibrosis is one disease that's caused by a lot of different potential gene changes, and so I think that there's certainly hope for some of these neurological diseases. There's a lot of research going on for non-genetic therapies for these.
I'll also add that just a few years ago, because of the lack of research in this area, the National Institutes of Health launched what's called the Brain Health Initiative. And it's a multi-million-dollar project to try to promote research that hasn't been done on brain health, neurological conditions. I know Cleveland Clinic and Case Western have some of those grants. And part of it was a reaction to the fact that it's been years and years and there hasn't been much progress. So I guess I'll be somewhat hopeful to say that my hope is that this new project, this Brain Health Initiative, will actually lead to better research on specifically neurological conditions like ALS, like Parkinson's, like all the various kinds of things that just have been largely understudied. So
We have time for one more question.
I was given a 23andMe kit last Christmas, a year ago. I have not found the nerve to go ahead and do it. Have you done this? Would you recommend that your sisters, brothers, mothers, fathers do this? Or is it still I'm worried that my information is gonna fall into the wrong hands. I have Parkinson's that runs in my family. Will I ever be denied healthcare coverage because I knew at some point that I might get it, etc. I mean, I think that is probably a question for a lot of people. Is this advisable at this point in the game?
I'll say 2 things. I really do think it's a personal choice. I think as long as you know what the risks are going in terms of the ways in which the information is used. We have not heard any stories, right? There's a lot of concern about genetic discrimination. I can tell you that there's very little evidence that a lot of genetic discrimination goes on in this country. Very little evidence. We don't see a lot of court cases. We don't see a lot of things of where people are denied insurance based on their genetic information. It happens, but we don't see a lot of that happening. It's really more about kind of knowing the limits of what that information gives to you and knowing the potential risks of, well, now I know this and I didn't want to know that information, right? Or now I know, and most of what we hear are things like that. I didn't realize it was going to give me information on my risk for X, and now I'm really worried about it. Now I got to go to my doctor. So it's more about whether or not you're prepared to get information that might raise worries for you. It depends on your concerns about the idea of other people, like pharmaceutical companies, using your information for research. Typically, that's not going to get back to you or harm you physically, what we call welfare harms, but there are non-welfare harms. There's the idea of privacy. And so it's really up to you about whether or not, what kinds of, how you feel about having that information out there. I can tell you that most bioethicists, including both of us, don't do it. Right, wouldn't do it. It's just being honest about my own. It's just not something I'm interested in. I do worry about the idea of other people having my information. I think it'd be kind of fun. I think it'd be interesting to learn about my ancestry. I think if I knew that I could do an ancestry test and not have that information used, I probably would do it because I'm interested in learning more about where my ancestors came from. But I worry about these other things, and so that's just my own personal opinion. I'll let Marsha.
Having said that, lots of geneticists actually do these tests, and they enjoy it, and because they feel like the information is their business, that's what they want to know. And the other thing is that a lot of people who are in 23andMe participate in forums on the 23andMe website that are specifically for research into these particular conditions, and they feel like they're doing really great things. And there is some really interesting research coming out of the 23andMe genetic databases because it's so huge now that it's enabling the kind of research that you can't do on smaller data sets. And so there is some really interesting research going on infant mortality, on certain kinds of heritable diseases. There's fascinating stuff going on, and a lot of people feel like that's really of value and they want to contribute to that. So having poo-pooed them all through this talk, I'll say that some people find a lot of value in it, and that's absolutely fine. We don't say, oh, you're terrible for doing that. It's up to you.
That's right. And research is good. We want research. We want research into things, right? It's less about the research being bad or good. It's more about control. And about your wanting to control what happens to your own information. That's really what it's more about.
On that, I think we'll end, and I'd like to thank Aaron and Marsha for a wonderful explanation of this important topic. Our next forum will be on the 26th, and the topic is Brexit, financial, U.S. and international impact, with Professor Elliot Posner, CWRU Chair of the Department of Political Science. And we welcome everybody to attend the service upstairs today at 11 o'clock, which is Superhero Sunday, and is presided over by Reverend Joe Cherry and Karen Tooley and Dr. Alan Georgia, Pat Solomon, and Dawn Arrington. So we welcome you to come upstairs for that. Thank you very much.
Mark told me your grandmother.
Yeah.
When was
I've got
the baby?
pictures, and then I've got to go. Then I've got to go to the ladies' room, which means the 7th, January 7th.
Oh, okay, because you were in Oregon or where?
Before we were in Oregon before that, and we're going at the end of the month. We were there in December.
When she was pregnant. You had to rush to make him to.
No rush. We have lots of time.
I want to go to the ladies' room.
Well, good genetics. Thank you.
Two smart parents.
Yeah.
This is David?
Yes.
Oh my God.
And of course, the baby has got to get into reading because that's what this family does.
Reading?
Yeah.
Teachers reading?
No, we just all read books all the time.
Oh, okay. Okay. Very lovely. And you know what? One time it occurred to me, it dawned on me at one point. You know, if you're not a parent, you can't be a grandmother. It's very disappointing.
Well, that is true. You have to adopt grandchildren. That's not easy to do. Yeah, it's not easy. We tried adopting grandchildren, just
sort of casually. It didn't
always work.
I didn't work for that agency. But anyway, well, I'll see you later.
Okay, we're going to go to the ladies' room.
The holidays are
over.